So our next speaker is Dr. Mike Wood, who's the CEO of New Farm. Mike did his PhD at Duke University, postdoc in neurobiology at Harvard, went on into industry to work for Lilly for a while as a postdoc, and found his calling there, went to AstraZeneca, where he led the psychiatry research initiative, and recently has been the founding CEO of New Farm. I've had the opportunity to work with Mike on a variety of projects. There you go, Mike. And put this around your head. Okay. Very good. Great. Welcome, Mike.
Thank you. Thank you, Mike.
Clicker?
Yep, I do. Thank you, Mike, and thank you, Hal, Tor, also. Am I? Nope. Oh, I see. I just didn't. So it's not that around behind you. Oh, I see, like this. Okay, here we go. All right. All right, now we're good. All right, thank you. Thank you, Mike. Thank you, Tor, for the invitation, and Hal for all of your help. And sort of listening to the previous talk, I thought we probably should have been flipped around, because what I want to do is just provide a little past as prologue for CNS drug development. And I thought what I would do is actually just basically review what's happened in the field maybe in the last 5.5 years or so. So this is just an analysis that I put together that's basically pulled out of CenterWatch, which reports all of new drug approvals. And within the field of CNS, between January of 2011 and I think I did this about a month ago, so I stopped at July of 2016, there were 61 approvals. And that actually represents 49 different brands. So about a dozen brands were approved for 2 different indications, or maybe it was 11, and there was one that was approved for 3. And then from those 49 different brands, I also wanted to point out that the FDA has an annual publication that they actually did start in 2011, so that was a good time to start the analysis, where they report out what are actually novel drugs. And essentially what this means is this is a new drug substance. It's not something that's appeared in the marketplace before. So again, of those 61 approvals, about 17 are novel, and I'm going to drill down a little bit further into those. And just for comparison, within oncology, about a similar number of approvals, maybe 10% of approvals overall versus 8% for CNS. More brands, considerably more brands, and a lot more novel entities coming out in that 5.5-year time frame in the CNS space in the oncology space versus CNS. So again, it's not a huge fraction in the last few years. CNS drug development used to be pretty dominant overall, and in this case, CenterWatch actually has a lot of different classifications, so I had to kind of jumble some things together. Oncology is a standalone, family medicine is a standalone, but the way this is reported, actually family medicine approvals that appear in the CenterWatch list may show up in maybe infection, and they may show up in other, this is a big jumble of immunology indications. But the major point is I actually think this is much lower than it used to be, say, 20 years ago in the days of olanzapine, fluoxetine. And it actually represents far fewer sort of novel drugs. The other thing I wanted to point out here, and actually the novel ones are here, they're the ones that are in the blue coloration here. The other thing I wanted to point out is, well, where are these drugs actually coming from? So this is based on 2016 data from the Fortune company, the sort of most gold Fortune 500 companies, and sort of slightly lower. These are sort of in the Fortune 500 to 1,000 range. Endo is in that Fortune 1,000 to 2,000 range. And you can see about 60% of drugs are actually coming from companies that are not even in the Fortune 2,000. So the major pharma are not really dominant in the CNS space in terms of the new approvals. And the other thing to sort of keep in mind is sort of go back to this idea that there's 49 brands of which only 17 are novel. What that really means is that about 32 of these are some pretty old drugs. Sumatriptan, for example, it's just a new formulation, for example. There are lots of combinations here, memantine, donepezil. So the innovation is in either nailing two things together that haven't been nailed together before, or it's in providing a new delivery mechanism. And these are not necessarily negatives. I mean, those are actually improvements in terms of treatment. I just wanted to place some context about what's going on or has gone on in the CNS space. Finally, because it's a sort of a mini passion of mine, what I also wanted to do is just point out that a number of these drugs are actually, their origin is in the academic space. So literally academic laboratories invented everything that you see that's sort of in red. And that's roughly 20% of what's represented here. So these are the 17 novel drugs. And I actually wanted to break these down a little bit further. So brivaracetam really is a novel drug. It's distinct. However, it's a follow-on from leveracetam and the whole racetam family, which has been around for a very long time. There's greater understanding of how these drugs work now, and that actually, I think, did drive the innovation around brivaracetam. But I would argue this is not a new mechanism, right? And this one maybe is probably the weakest of this argument. The aripiprazole, Lorexol, that's a prodrug. So it's an intramuscular formulation of aripiprazole. I would argue that both brexpiprazole, cariprazine, these are both analogs of aripiprazole in many ways. They are D2 partial agonists that have some other ancillary pharmacology. I can say this now because I don't work for AstraZeneca. Nalexidol is really just it's a pegylated derivative of alpha naloxone. Lots of these others are, the melatonin agonist. I mean, there's been agamellatine and at least one other prior to this. So I would argue that of these 17, really only 5 are new mechanisms. And so pimavanserin, which I think is a really interesting drug, I mean, in particular because 5-HT2 selective agonists were pursued for schizophrenia years ago by Mira and Merrill Dow. Didn't show efficacy in the clinic, and so pimavanserin's now really focused on psychosis, but in PD patients, and I think that's kind of an interesting past as prologue as well. But the others, suvorexant, droxidopa, arguably that's not all that new. It's been in Japan since 1989. It's just new to the US market. Perampanel, that's an interesting one, I think. And then retigabine or ezogabine, the KCNQ channel opener. These are all new mechanisms in the CNS space within the last 5.5 years. So what are these treating? Well, as I mentioned, PD psychosis. That may be, in some respects, the most profound innovation with a new mechanism, I would argue. But a couple of new anti-seizure drugs. This is actually a fairly swamped space from the commercial perspective. Neurogenic hypotension, is that really a CNS indication? I think that's arguable as well. Insomnia, again, that's starting to get to be a pretty crowded space as well. So my argument here is we're really not solving the big problems in CNS with these new mechanisms. So pretty somber picture overall, and I'm sorry about that, but these are facts. So what I wanted to do from here is sort of pivot towards what this conference is about and how that's actually going to impact. And that's why I think we probably should have been switched around, because I thought that was a beautiful example of what precision medicine could be doing to treat a bigger problem than neurogenic orthostatic hypotension. But we need improvements in disease models. You know, and this is kind of a broad definition, I think, for precision medicine. I'm not sure everyone would agree that, you know, improving disease models is precision medicine, but from my perspective, this is a major issue. One of the, and you think back to Dr. Miller's presentation, he had the failure rate and how that was increased with biomarkers. Actually, if you just look across indications, CNS indications have the worst failure rate in phase 2 clinical trials, right, when you actually go to test the concept. And in part, I think that can really be lined up against, that can really be lined up against the fact that there's very poor disease models for a lot of CNS diseases. So better identification, better validation of therapeutic targets is going to address probably that biggest issue. We do need improvements in biomarkers. I thought the previous talk was an excellent example of how those can be driving testing of novel concepts much more quickly. I think one of the things that the pharmaceutical industry does poorly is we're sort of, you know, the industry is really lined up to push to the market as quickly as possible. And what that oftentimes means is don't take time to do clinical research and learn whether or not your therapy is going to have a positive effect. Did you actually identify the right patient population? And to do that in a smaller setting with more precise measurements like proof of mechanism, you know, target engagements, you know, sort of the buzzword for CNS. That's really important because you can do this in many other disease areas, but you need, you know, if you have a PET ligand, at least you know you're testing your hypothesis when you get into a clinical setting. I actually think some enhanced accuracy in diagnosis and stratification. Dr. Flint's talk on depression was really captivating. My question coming from, you know, looking at those data is, this is a vast disease. You know, I've seen Michael Thace talk multiple times about depression and how there's many different axes that you can sort of slice that disease. Is this, you know, the genetic underpinning that were identified in that study, does this represent a specific subset of disease? Can we target therapies around that group that maybe harbor that mutation? And those are the types of advances that would really reduce the failure rate, I think. And, you know, sort of similarly, once, you know, this is purely thinking about the research and development component of it. Once patients actually are being recruited, are we recruiting sort of the population that we need to test the hypothesis? Again, the pushback on that from the commercial folks is we don't want to limit, you know, if you're trying to bring a new drug forward, you don't want to limit who actually might be prescribed your new treatment. So the tendency is to not be too narrow in defining inclusion-exclusion criteria for your clinical trial, because that just means that you get an approval for a very narrow subset of patients. However, I think there needs to be some realism layered on top of that and sort of focus in on trying to test that hypothesis. And then finally, you know, this is sort of again, another generic concept, but better, you know, improvement in dosing. And, you know, this can be adherence, it can be better understanding of PK, PD. We oftentimes just, you know, historically, you know, was maximum sort of tolerated dose without seeing some adverse events and go forward, not knowing whether you needed to actually treat. You had to have you had an AUC effect, so you needed to have drug on board all the time, or is this something that maybe a treatment once every 5 days would actually be more efficacious than something that was on board all the time. And so that's it. And just wanted to leave with you know, this is a collaborative effort because these are big problems. And so I wanted to leave with some collaborating mammals. So.